Hello, everyone. Thank you to participating to this ESCMID webinar related to the diagnostic COVID-19. With Jacob Moran-Gilad, we are very happy to receive you to this webinar. This webinar will be composed by 4 talks on the most important aspect of the diagnostic of COVID-19, in particular in the vaccination time, such as the moment. We are 4 very nice, very important speakers who will stress all the aspects of the diagnostic for this important disease. So it is a great pleasure for me to introduce the first speaker, Gilbert Greub, from the CHUV in Lausanne, Switzerland. Gilbert is a very important clinical microbiologist with a great experience of molecular diagnostics, and it's a great pleasure for me to introduce you to this talk about the molecular diagnostics for COVID in the vaccine era. Please, Gilbert.
Thank you very much, Maurizio, for introducing me. My talk is entitled Molecular Diagnostics for COVID in the Vaccine Era. This is our institute, this old yellow building, and we have also part of our laboratory in the hospital. So we are based at the University Hospital. And I will just provide just one slide regarding my field of interest. I'm medical advisor of Resistel, but it's about antibiotic accessibility and nanomotion. And I have also research agreement with Resistel, Suez, and BD, as well as I'm co-director of JeuPro, a game company. So I think that you are all convinced that laboratories are pivotal to control the outbreak by detecting cases and allowing isolation and quarantines. In Lausanne, we have done since 14th of February 2020 more than 140,000 RT-PCR, and currently we are still doing about 40 PCR per day. And this is important in order to stop the spread of the SARS-CoV-2 that you see on that cartoon. And this represents with the microscope the labs and all the diagnostic tests. But as you know, we have also some reagents difficulties when the borders have been closed from time to time, especially during the first lockdown. The gold standard for RT-PCR is nasopharyngeal swabs, and due to problems with shortage in these thin swabs, there was some large swabs that have been done during the early days of the outbreak. By chance, now we are back to the normal nasopharyngeal swabs, and we can do a proper sampling that is associated with a really good sensitivity of about 96 to 98%, at least in our institution. And also in our institution, due to high level of homemade automation or to the automated cobas system, we have a specificity which is really good, significantly higher than 99.9%. We have also a relatively short turnaround time, and I will discuss these different aspects in the next slide. Of course, we are not only using this type of high throughput machine to do high number of tests per run, but we also move to some rapid diagnostic tests, just as the Liat from Roche, which allows you to have results in 20 minutes when you insert the samples in the machine, and also the GeneXpert has a relatively low turnaround time of about 45 minutes. The RT-PCR is really important for epidemiologics to track chains of transmission and to confirm cases. And this provides us with all this epidemiological data that we follow on a daily or weekly basis. It is also important in the hospital to make a proper orientation of the patient in COVID area, and also to test asymptomatic patients that enter the hospital in order to keep a safe hospital. The cutoff for contagiousness is discussed. Maybe less than 1,000 copies per ml is a negligible amount of copies for contagiosity in the hospital when the subjects are in contact for prolonged time in the same room, and likely between below 10,000 copies per ml is a lower cutoff for contagiousness in the community. Regarding patient management, RT-PCR is also pivotal to provide the diagnosis of COVID in symptomatic subjects, to allow screening in asymptomatic subjects, to provide to look for alternative etiologies, to better understand the timing of the disease, and also to allow some insight in the effect of antivirals or immunomodulation on the viral load. In addition, it was pivotal to have RT-PCR tools that are amenable on different types of samples to allow the description of new clinical presentation, such as meningoencephalitis or miscarriage. I want to first to convince you that despite we have done a lot of samples in 2020, from 14 of February to 31 of December, we processed 128,62 samples. And despite this huge number of samples, we keep a short time to results. And practically, you can see here on this slide that we had most of the samples that we have results with less than 6 hours from the time the sample arrived in the lab to the results on the iPhone or the phone of the person that is tested. And we provide the result by SMS to the person tested directly. So 64% receive the results in less than 6 hours, 86% in less than 12 hours, and more than 99.4% in less than a day. And of course, we were not as good initially because we had not an automated validation, and we moved to automated validation in April. And from May, as you can see here, we have a relatively better performance with some months, especially recently, with more than 75% of all results available in less than 6 hours. Overall, a short time to result does not mean a poor sensitivity. The sensitivity is of about 96 to 98%, as shown of 4 different studies that we have performed. The first is a paper from Muller about the automated validation, and the 3 additional ones were all PCR saliva versus nasopharyngeal PCR, and you can see that we had a really good sensitivity when considering the saliva PCR as the gold standard for the nasopharyngeal results, from 95 to 98%, and a fair sensitivity of RT-PCR on saliva when you consider the nasopharyngeal PCR as gold standard, with a range from 69% to 95.7%. You may be surprised to have a solo sensitivity with saliva PCR. This is due to the fact that in these hospitalized subjects, we have a median number of days since onset of symptoms of 11 days. And this makes a lower viral load because you have already, you are not at the acute phase. And this is really important also when you consider antigen tests, which are not the topic today. But for instance, in the first study that we have done with Schwab et al., we had 85% of sensitivity for antigen test as compared to 95.7% for nasopharyngeal PCR. And now, using the Roche antigen test, the same test that was used by Schwab et al., with Antonios Kritikos, we found in our hospitalized subject only 35% of sensitivity using the same approach. And so it means that clearly nasopharyngeal PCR and saliva PCR are much better than antigen tests in terms of sensitivity. We know that the analytical sensitivity is about 1,000 to 10,000 better for PCR as compared to antigen. And this high sensitivity of PCR is also observed in pediatric subjects with 95% in nasopharyngeal samples and 85% in saliva samples. The sensitivity, of course, depends on the symptom duration, and this is the viral load expressed in CT according to the days since symptom onset. And you can see you have a much lower viral load during the first 4 days of symptoms, with a median of about 24, 25. And this median of CT is up to 34, 35 in subject with more than 16 days of symptoms. So practically, you see also that the symptom duration, this is overall in our patients, and this is another project in pediatrics, and so in subjects between below 18 years old. And you can see that the viral load largely decrease in the nasopharyngeal samples, but is always relatively low and about at the same amount in the saliva according to days of symptoms. Not only there is a short turnaround time, not only there is high sensitivity, but there is also an excellent specificity. So for instance, in a small retrospective project that was done at TUBE in our hospital on a study about respiratory tract viruses not related to SARS, and that started already in November 2019, we analyzed in our lab retrospectively all these samples, and all of them were negative. And you see there is quite a number of negative samples before the first detected Swiss SARS-CoV-2 case that occurred on the 25th of February in Switzerland. So before the outbreak, on samples taken before the outbreak, our PCR was always negative, despite at the same time we processed quite a number of highly positive samples with millions of copies per ml. So this is the first reason why we consider our specificity excellent. A second thing is that after the peak of the first outbreak, we have seen several days with no positive samples, especially in May and June, during a time where we had really low circulation of the virus. And the third thing is when you look at the location of the negative and the positive test, the negative are in black and the positive are in red. You can see, for instance, in this area, which there is a lot of mountains here, there is no circulation of the virus during all the first wave of the outbreak, and we have only negative results. So it means that when there is no circulation before the outbreak, or in location where there is no circulation of the virus, we have no positive results. Of course, in towns such as Lausanne, Morges, Lyon, there is quite a number of positive cases as expected, and we identified clusters. A question that a lot of medical doctors or lay people ask, why are you still using 45 or 50 cycles? So in our lab, with the cobas test, we are using today 50 cycles. And of course, some of my colleagues, such as Valérie Dacremont, said that it's insane to use so many cycles, 25 or 30 cycles would be sufficient in terms of epidemiology. And she does not consider that patients are contagious above this number of cycles. But if you look carefully of what we have observed, is that among the first 3,000 COBAS tests that we have done that were positive, all were positive, we had only 4 subjects that had positivity after 40 cycles for the E gene, and only 214 after 35 cycles. So it means that it's really a low number of positive. And moreover, these 4 samples that were above 40 cycles were all between 40 and 42 cycles. The same for the RDRP gene. We did not observe any positive after 40 cycles and only 14 above 30 cycles. So it means we have really few numbers of high CT values. So most of the time, the fact to go to 45 or 50 cycles does not add any problem. On the contrary, to see that there is no positivity above 40 cycles allows you to be confident on the quality of your probes, that there is no disintegration and release of fluorescence due to a probe that will not be correct. So I redo just yesterday all the stats, and now we have 9,000 COBAS tests, and we have no more 4 but 7 above 40 cycles. So the ratio is even better than what I present here. And in addition, we need to go behind 30 cycles because, as you can see from this paper from the Marseille team, between 29 and 33 cycles corresponding to 10 to the 4 to 10 to the 5 copies per ml, you can still recover the virus in culture in about 56 to 78% of the cases. So it means that between 30 and 35 cycles, so until 1,000 copies, the patient can still transmit from time to time. And in the spatio-temporal evaluations that we did, we looked at different clusters. And what we observed is that we observed a total of 20 clusters of 3 cases. All 3 had a number of copies at time of diagnosis that was lower than 1,000 copies per ml. So all these clusters, these 20 clusters, would have been missed if we have not done and used the results which are above 30 cycles. So it's completely stupid to stop at 30 cycles. That's what I think, and that's what I say as well. Practically, the problem is that below 10 to the 5, between 10 to the 4 and 10 to the 5, what we observed, at least in the population, in Switzerland, in Swiss population, there is still a significant contagiousness. And this contagiousness was mainly observed in the elderly because these subjects, when they get the infection, they don't get it in the metro, they don't get it just by smoking with colleagues, so not due to high number of viral load, but really when they get in close contact with their family and they just trust that their family are negative and non-contagious and they get infected even with low number. So it was really impressive to see the difference in terms of contagiousness in cluster of older subjects or younger subjects. Younger subjects were cluster where you see subjects with very high viral load of more than 1 million copies per ml. practically, I had to discuss about PCR in the vaccine era. And of course, what I have showed you is all the diagnostic molecular platforms, the COBAS, the rapid tests like XPERT, BEMAX, and COBAS, and all these tests that we use since February and progressively implemented additional ones allowed us to know what happened in our location with the first and the second wave of infection. And of course, with this reliable test, we have a good idea of the epidemic. In late December, we faced the second problem, which was the problem of the UK variant and South African variant and Brazilian variants. And due to that, we had also to adapt and to implement a S dropout PCR to detect using Thermo Fisher kit the possible UK variant, and also implement a PCR targeting specifically the 501 mutation, and also to use our genomic facility not only for research on the variants of the viruses, but also to do epidemiology. So this is what I want just to stress in two slides. So the first thing is that we use this QuantStudio 7 PCR to look for variants that have a tyrosine at position 501, and also we used as others genomics to look at the spread of the virus. And what we have seen, this is the data of all the University Center of Switzerland. Goncalves and Adrian Negli has piloted this project. You can see that there is quite a number of UK variants in this from Lausanne in our area. And of course, you can see all this positivity from the western part of Switzerland and for the northern part and the German-speaking part of Switzerland. So in conclusion, PCR tests are very robust, and I think this is the most important. People should be confident in what we are doing, and this is a message that I focused on our own experience, but I think it's just an example how we should communicate for our own labs. And it also provides material for search of the mutation or for whole genome. And in the era of variants, we need to go back to PCR, and we have to change the paradigm. And especially the antigen tests are only good during the first 4 days on symptoms and have major drawback after 4 days of symptoms in terms of sensitivity. So the PCR should be a priority over antigen test, and antigen test should really be limited to these 1 to 4 of symptoms, because after that, they have a really low sensitivity. And in our spatiotemporal analysis of cluster, we would have missed 24 clusters if we would have used antigen tests instead of PCR during the first wave of the outbreak. Thank you very much. This is just a nice area where our hospital is located, and this is what we can see from the window at the top of the building of the hospital. So I'm available for questions. Thank you very much.
Thank you so much, Gilbert. Thank you for this very nice panorama. I know very well, it's a fantastic location. Okay, I have, first of all, I have a question from the audience. Now, 2 questions. The first is related to the use of CT for the evaluation of viral load and general infectivity. Is there a better way to determine the amount of virus other than CT? And if we use the CT, is possible to normalize this value?
Okay, so practically in our institute, what we do is we transform the CT value in viral load. And so we have used different equation according to each test, because the different machine instruments have not the same results. So you have to adapt because you have also to adapt according to instruments. So we have done, we have used IFU, so virus quantified in culture, in order to derive the proper equation to be used for COBAS viral load quantification in copies per ml. So my suggestion is that we all move now to copies per ml, which is much more clear. And you have seen that in Fougère et al. Paper, for instance, I have shown copies per ml. And practically contagion is really low, below 1,000 copies per ml, and is, to my opinion, negligible below 1,000 copies.
Okay, thank you. We have a lot of questions for you, Gilbert. So the second is Switzerland considering using antigen tests in school or not? You have some information on this.
So practically what we decided now in our Canton de Vaud with the public health officer is to focus only using saliva PCR at schools because you can use saliva sample, and with that you have good results. We have done a study that is not yet published, but we got using the same good antigen test on saliva only 4 to 8% sensitivity. So if you use nasopharyngeal swabs for antigen test, you have 42 to 35% of sensitivity. If you use the same antigen test but on saliva, you go down to 4 to 8%. So if you use antigen, you need to take a nasopharyngeal swab. And I think for asymptomatic subjects, especially children, it's not acceptable to be so invasive. So it's much better to do saliva PCR. And then there is the question of pooling. Some people say you have to pool, it will be cheaper. I always think that what is cheap is at the end more expensive. It makes a lot of burden, a lot of work. And finally, if you pool all the sample from a class, and then you have a single positive, you have to go back and resample. If you start to make the pool reversible, it's a lot of work, and it's also a risk of contagiosity if you do that at the class. So if you do that in the lab, you can have contamination. So I think, and at least that was the decision locally, we go for individual saliva PCR.
Thank you. Only another question, and then if you, I asked to William to collect all the questions and send to Gilbert for reply, because we have not the time to reply to all these questions. Sorry. This is a very burning question, in my opinion, at least from my experience here in Italy. How do you interpret the samples that are low positive only for the E genes? For the E gene, but negative for other? This is a very burning question, Gilbert. Me too, I'm interested in your reply. Thank you.
So among the 3,000 samples, we had 12% of only E gene positivity. We know that the E gene is slightly more sensitive than the RDRP for a variety of reasons, and especially also due to the biology of the virus. And so I always think that they are true positive. And one of the reasons why I think it's true positive is, for instance, I have two, I called one of these ICT value above 30, above 40. This guy was above 40. And I asked, what happened, blah, blah, blah. And why have you been tested? And he said, oh, I'm asymptomatic. I just have rhinitis 2-3 weeks ago. And I said, but why have you been tested? Oh, it's just my sister is working in a home for elderly persons. So that's why she was tested, and she was tested positive. And then subsequently, I was tested because I was a contact. And her sister was also low, also above 35 cycle, and practically both of them were very low. Of course, due to the presence of variants, it is also possible that when you have a single positive PCR, you have mutation in the target of the other PCR. So you have also to take that in consideration. So if we have more than 6 cycle difference between two targets, we do a whole genome. But of course, we cannot do a whole genome if there is more than 34 cycle, despite we have a good library preparation approach. I know others only do genome until 30 or 32 cycle. In our hands, we can go up to 34, but above 34, we have really a low coverage, and it's too dirty to be used.
Okay, thank you so much, Gilbert. The other question will be sent to you for reply. Sorry for not able to reply to all these questions, but we are, of course, sometimes. Thank you so much once again. And it's a great pleasure for me to introduce the next speaker. Is Professor Mario Plebani from Padua, Italy, a great expert about serology in general, and in particular for our topic, the COVID serology. The Mario talk is related to the COVID serology before, during, and after vaccination. So Mario, this is a very important and very timely talk in this vaccination era. So please, and thank you for being part of this webinar.
Thank you, Maurizio. First of all, let me thank you for kindly inviting me. I thank you, the ESCMID Society. It's a great honor. I'm delighted to deliver this lecture. And you said that the title is COVID serology before, during, and after vaccination. But first of all, let me introduce my university. I'm full professor of clinical biochemistry and molecular biology, University of Padua. You can appreciate in this slide on the right side the magnificent Aula Magna dedicated to Galileo Galilei, who was teaching for 10 years in our university. On the left, the first and oldest anatomic theater in the world. So when the COVID pandemic will finish, I like to invite you to visit our university and to enjoy, to share with you some science after the COVID, please. So this is what I like to share with you today, COVID-19 serology before vaccination, the evidence we have, the issue of the relationship between serological assays and neutralizing antibodies, and the problem of the persistence of antibody responses. During vaccination, I like to show you some data about the role of serology in clinical trials. And finally, what probably should be much more important today is after vaccination, what we have to do for monitoring, and finally the issue of variants and the eventual effects on vaccine and neutralization activity. So first of all, I like to start with this meta-analysis published in British Medical Journal Evidence-Based Medicine. As you can appreciate, unfortunately, the risk of bias in publication already reported in the literature is higher, particularly for point of care and home care testing. I'm speaking about serology, but the risk of bias is lower, but is still present also for molecular testing. So now I like to introduce you the evidence supporting the application of serology before vaccination for seroprevalence studies in general population and high-risk subgroups particularly healthcare operators. We made something about this, some papers about this. The issue of contact tracing, very important. To identify donors of convalescent plasma therapy, intriguing and interesting field. To identify prior infections, the so-called late diagnosis, very interesting. And now to assess vaccine response in clinical trials and monitoring. Vice versa, no evidence has been collected to support the application to diagnosing an acute infection, to provide disease prognosis, to screen units of blood for SARS-CoV-2. So for acute infection and other, only molecular is the right tool. And we have to collect much more evidence to identify people to vaccinate with priority, and therefore to guide to vaccination strategies, as guide to vaccination strategy, to predict susceptibility to infection, reinfection, and to infer immunity issue, the so-called immunity passport. So wait for an answer to these issues. The main requirement for serological tests are performance characteristics, first analytical and second diagnostic, because each specific case shall be validated and verified by laboratory professional in cooperation with the in vitro diagnostics and clinician, of course. Only immunoassays with well-demonstrated correlation between antibodies, circulating antibody, both total and IgG, with neutralization activity shall be used. Immunoassay providing quantitative measurement shall be preferred, as semi-quantitative results are not enough. And finally, very important, appropriate cutoff for each specific assay shall be identified to allow the right clinical interpretation, particularly if the disease prevalence is not so high as expected. So before the use, in precision, dynamic range, or linearity, cross reactivity, the cutoff, diagnostic, sensitivity, specificity, predictive positive and predictive negative value, and finally correlation with neutralizing antibody should be verified. For monitoring, internal quality control as well as external quality assessment scheme should be performed. You see in the bottom of the figure the experience in our region with an external quality assessment scheme. And now this slide shows you what is well known, the type of target we can use to develop a serological test. And what I like to stress is that today, most of the new methods under development include the receptor binding domain as a target, or the so-called trimeric spike protein. And we are moving toward this type of new targets in the present assays. So very important is the relationship between serological assay and neutralizing antibodies, because it's clear that we are using as a surrogate measure serological test, but what we would like to know is that the role of neutralizing, the value and the concentration on neutralizing antibody. So the vast majority of SARS-CoV-2 infected individuals seroconvert within 5 to 10 days post-symptom onset, with about 95 seroconversion by day 9 and more than 99% of subjects develop an anti-spike antibody response. This is a reference, but there are many papers dealing with demonstrating and confirming this concept. Neutralizing antibodies develop more or less on the same time frame as a seroconversion. And neutralizing epitopes on the SARS-CoV-2 receptor binding domain appear to be highly immunogenic and easily recognized by antibodies. So these are evidence collected in the last few months. And this is a paper demonstrated that, in fact, some of commercially available immunoassays present a good correlation with neutralizing antibody. But in particular, a strong correlation between levels of receptor binding antibodies, receptor binding domain binding antibodies, and the SARS-CoV-2 neutralizing antibody has been reported by several groups, particularly in this paper, by Prem Kumar and colleagues. And you can appreciate in this slide the type of correlation that is really very significant, both for IgG against the receptor binding domain and also IgM. And we'll discuss a little bit at this point. This is in our experience with another commercially available immunoassay using as a target receptor binding domain. You can appreciate the statistical significant correlation with neutralizing antibody. Neutralizing antibody measured with the gold standard PRMT, and you know very well this technique. And this is another paper we have published demonstrating that even for other commercially available immunoassay, it's possible to observe a good correlation, not for all, with neutralization activity. So, in other papers, I like to stress that almost all analyzed the COVID-19 convalescent subjects produce neutralizing antibodies, can be detected at an early stage of the infection. And what I like to stress is in this paper, in another paper, neutralization activity is mediated not only by IgG, but even by IgM, and this should be better evaluated in the future. So the contribution of IgM and IgA needs further confirmation. Probably you know this paper by Klinger and colleagues. I show you the type of correlation with neutralizing activity. So another very intriguing issue is the persistence of antibody responses. Of course, is related to the issue of the reinfection and the rate of reinfection. I like to stress that the last published paper in Lancet on October last year demonstrated that up to now the evidence is about 4 cases of true reinfection, but probably will increase over time. But anyway, the problem is that we know now that the titer of spike IgG, SARS-CoV-2 spike IgG, are stable, and the half-life is 103 days. For the receptor binding domain IgG, the titer seems to be near to 83 or 4 or 19 days, very similar to the half-life of neutralizing antibodies. This is a paper recently, very recently published in Science. So I like to stress that the stability of the antibody response may also depend on the target antigen. So we have to select the right immunoassay or the right neutralization assay. And this is what I said you, the data provided in the paper by Jennifer, Dan, and co-workers. In another paper, very similar durability has been found in longitudinal assessment of antibody titers over 7-month period using both N and S antigen, using Abbott, Roche, and Diasorin. The half-life of IgG was found to range from 86 to 202 days. Weinberger and colleagues publishing Science showed that IgG and neutralizing antibody are stable for at least a period of 5 months, but I can assure you that we have data under publication with the Andrea Crisanti group demonstrating the persistence until at least 7 months of the antibody. So we know that probably we have 2 types of subpopulation. The so-called decayed, in which the anti-SARS-CoV-2 IgG decline soon, rapidly, but there is another subgroup with sustained anti-SARS-CoV-2 IgG. And I like to stress at this point that not only serological assay, but the humoral responses give us an answer about the durability of immunoresponse after COVID-19. So what about serological tests during in clinical trial for vaccine? You know that both for Moderna vaccine, they studied the response of the efficacy of the vaccination using both immunoassay, particularly receptor binding domain as a target, but also, of course, neutralization activities, demonstrated good correlation between immunoassay and neutralizing activity. And even the persistence over time of the antibodies has been evaluated using this type of serological and neutralization assay. The same for the other vaccine, particularly the Pfizer vaccine. You can appreciate the days after vaccination, the response with the serology and neutralization, but also they studied the immunological response. The same with the AstraZeneca. I can show you the type of neutralizing antibody titers and other immunoresponse. This is the vaccine BB152 from India, the same. And finally, the Sputnik, in which the humoral response against the receptor binding domain and neutralizing antibody has been well evaluated. This paper is published in Lancet very recently. So they used serological tests to provide evidence of the efficacy of this type of vaccine. And now the question is what happens after vaccination? We published this paper on it is a recommendation to use a serological test, but even other type of immunoresponse testing for providing evidence of the efficacy. And I will deal with this in a few minutes about the prioritization of population to be vaccinated. So this is just to show you what we are going to do to follow. This case are healthcare operators, that received vaccine, is a Pfizer BioNTech, and you can appreciate the serological response. But this is not new, of course, is what we are expecting. What about the prioritization strategies according to the age and status? So there are pros. Poor countries should maximize cost effort for reducing viral spread, reduce unnecessary potential side effects. Cons are pre-vaccination serological screening cost effects should be better evaluated. Effort for managing serological results and vaccination should be done. And finally, but very important, robust analytical methods tracking neutralization antibody levels are required. So I like to show you this data, very recently published. It's a preprint by Kramer and colleagues, demonstrating that is very important, at least in my mind, and as observed by this author, to evaluate the serological status before vaccination, because to avoid systemic reaction. And you can appreciate that seropositive subjects at the vaccination, in fact, developed systemic reaction, side effects, no mortality, but side effects. So it's very important to stress this point in my mind. Another problem, of course, today is the SARS-CoV-2 mutation and the variability, particularly in the spike glycoprotein that can affect the efficiency of antibody neutralization and the use of vaccination and vaccine. And you know, this is particularly true for the South Africa variant, in which we need more data to clearly demonstrate if really vaccine we have are able to assure protection or not. And the same, just another paper to show you what we are doing in several parts of the world to provide evidence of the neutralization of this type of variant. So, open issues are the type of targets according to specific vaccine design, that means which type of serological test we have to use, which type of subpopulation should be evaluated, different age, but particularly immunosuppressed patient, subjects and patients under special therapies. We need to identify the titers which assure true protection. This is not an easy task. And we have to better integrate serological data with cell-mediated immunoresponses. And I believe this point is very important. So my take-home message are SARS-CoV-2 antibody testing plays a key role in improving, first of all, our knowledge of the type of immunoresponse to this new and still mysterious virus. So it's very important seroprevalence surveys, epidemiological surveillance, and today, not only in vaccine trials, but in monitoring the antibody response to vaccine. Only well-designed, validated, and quantitative assays should be used, and they provide a demonstration of good correlation with neutralizing antibodies. Further studies are needed to better evaluate the persistence of antibodies over time, and of course, the type of cellular immunoresponse. SARS-CoV-2 antibody testing have been used and shall be used, therefore, in clinical trial to evaluate the efficacy of vaccine and possible effects right now of virus mutation. This is very important. SARS-CoV-2 antibody testing should be used to screen individuals prior to vaccination, particularly if infection story is unknown, and we know that for many people is unknown, to reduce the risk of systemic reactions, the so-called reactogenicity, and to evaluate the immunogenicity after the first or second dose. And finally, systematic population studies are required to determine the full extent of protection provided by previous infection and vaccination. So thank you so much for your attention. Thank you so much for giving me the opportunity to deliver this lecture. This is my email, and for any question, please do not hesitate to contact me. Thank you so much, Maurizio.
Thank you so much, Mario, for this very comprehensive and complete presentation about this very burning topic. We have some questions for you from the audience. And the first is, do you think there is a risk in developing all the serological assay to the spike antigen, since this is also the mutation site in variants of concern? So there is the risk that the serological assay cannot be so much sensitive in patients infected by variants?
Up to now, and with some variant, particularly the UK variant, this fear is not so evident. But in the future, we have, of course, to evaluate the new variant and to provide evidence that the new immunoassay, particularly using as a target a receptor binding domain or trimeric spike protein, should not be affected by the risk of mutation blocking the reaction. So it's a moving target in my mind.
Yes, I fully agree with you, and we have to be careful about the results. So another question is about IgA. This is also, I combine the question from the audience with my personal question. So the question from the audience is, do vaccines lead to the production of IgA? And my question is, do you think that IgA are a good marker for monitoring infection by SARS-CoV-2? Because we have some experience in some very early production of IgA. So what do you think about it? Of course, IgA found in the serum, not in the saliva, of course. What do you think?
I think that the role of IgA is unfortunately underestimated, and we have to provide more evidence about the value of IgA measurement. We started to evaluate IgA in saliva, but I still believe that further studies are needed to provide some more evidence of their role in protection, and not only in saliva, but also in serum or plasma. So I totally agree. That is a field of research. We have to give more efforts to provide much more studies.
Evidence, yes. A final question, and it's not really a question for you, I think, but it is another important point. What do you think about antibodies therapy? I think that the question is related to monoclonal therapies or plasma convalescent therapies against the new variants. In your opinion, there is possible to use an antibody therapy for patients infected by SARS-CoV-2 variant or not? I know that this is very burning question.
This is a hot question, and you know, there are discussions even before the mutants, the variants, about the role of particularly convalescent plasma. I like to say that in Padova we have good experience regarding the use of convalescent plasma, and I believe that even for convalescent plasma and monoclonal antibodies, the answer is the time of the infection. So an early infection seems to be associated with a good response to this type of convalescent or monoclonal antibody therapy. In the future, I like to say that not with the convalescent plasma, but with the monoclonal antibodies, recognizing the variant, we should do a good job to care the patient. So I'm quite optimistic about this.
So a final information for you, if you can reply to a more personal question in the R&D, and so we can move ahead. So thank you once again, Mario, for your presentation, your very comprehensive reply to questions. So now we can move to Jacob Moran-Gilad, my co-chair, to proceed with the second part of this webinar. So please, Jacob.
Thank you very much, Maurizio. Mario, can I please ask you to stop sharing so we can move to the next presentation? Thank you. And I'm delighted to introduce our next speaker, Bali Pulendran. Bali, who I'm sure most of you know, is a professor of immunology at Stanford in the US. And for many years has been doing research on systems immunology and vaccinology. And he has now also very interesting data related to COVID. And so please, the floor is yours. Thank you for being with us. And the topic is laboratory assessment of SARS-CoV-2 immunity.
Okay, great. Now I'm unmuted. Thank you very much, Jacob. And I would like to thank you and the organizers for the invitation to this very interesting symposium. And I would like to tell you about work we're doing in my laboratory at Stanford to understand the nature of the immune response to COVID in humans. And the approach we take, as Jacob mentioned, is a systems immunology approach. And for those of you who are not experts or not specialists, basically what the systems immunology approach involves is to analyze the immune response at multiple levels of the biological hierarchy. So at the cellular level, you can use CyTOF, which is a technique that allows you to measure the dynamics of cell populations in blood, human blood. And you can use up to 40 markers, phenotypic markers, to define the various cell types. You can use RNA sequencing or single cell RNA sequencing to measure the transcriptome. Now there are multiplex cytokine platforms such as Olink that allows you to measure cytokines and chemokines in the plasma. Then using mass spectrometry or other platforms, you can measure the metabolites and the antibody response and the T cell response. So right at the beginning of the pandemic, now almost a year ago, we were very, as many labs were very interested to find out what happens to the immune system when this virus enters the body. And remember, this is a brand new virus and nothing was known about the host response to this virus. And so we were very fortunate. Back in February of 2020, we were able to establish collaborations across multiple sites, both within the United States, but also internationally, to have access to samples from people who had been infected with SARS-CoV-2. And so the question was, what is the nature of the immune response to this virus? Is there an immune signature that can predict disease severity? If so, could we have a diagnostic marker that can predict disease severity? And what is the immune response in asymptomatic individuals, considering that the majority of people develop hardly any symptoms or no symptoms at all? So this was essentially what we were interested in doing. And some of this has been published. This was a paper that came out back in August of 2020, and I'm going to show you some data, but this work is ongoing, and I'll show you some new unpublished data as well. So the initial work involved analyzing blood samples obtained from our collaborator at Emory University in Atlanta and from our collaborators in Hong Kong. This was in the laboratory of Professor Malik Piris at Hong Kong University. This was from Professor Nadine Rupael at Emory University. And so let me show you some data with the CyTOF analysis. And what we saw, and this is a Tisney plot in which you can see all the different populations of cells in the blood, B cells, basophils, T cells, and so on. And now this has been confirmed by multiple groups, but it was very striking that in acutely infected COVID patients, there was an increase in the frequency of plasmablasts. These are the cells that make antibody, and an increase in the frequency of effector CD8 T cells. That was what it looked like in this cohort from Atlanta, and very similar results in this cohort from Hong Kong. Again, increased frequency plasmablasts and effector CD8 T cells. What we also found was that there was a reduced frequency of plasmacytoid dendritic cells. These are the cells that make interferon alpha in response to viruses. And this was true whether we looked in Atlanta or in Hong Kong. And interestingly, in our CyTOF panel, we had an antibody that could stain or that could bind to this phospho-S6K12 kinase. Now, some of you might know this PS6 or phospho-S6K12 kinase are downstream mediators of mTOR signaling. And what we found was that the expression of this PS6 was diminished in the plasmacytoid dendritic cells in COVID-19 infected patients, either from Atlanta or from Hong Kong. Now, why is this so important? This is important because in a previous study that we had done 10 years ago, more than 10 years ago, we had shown that this molecule, P6K1 and 2, phosphorylated form of the S6K1 and 2 kinase, which is a downstream mediator of mTOR, was absolutely essential for the elicitation of interferon alpha beta in plasmacytoid dendritic cells. Now, of course, that study was not in the context of COVID. COVID wasn't known back then, but it was in the context of TLR ligands. But nevertheless, it was striking that now in COVID, we see that the expression of this marker was diminished in plasmacytoid DCs, and it raised the obvious question, well, does it mean that the plasmacytoid DCs in these infected patients are impaired in their capacity to secrete interferon alpha beta? So we did the experiment. We isolated PBMCs from infected patients here, or from healthy people. And without any ex vivo stimulation, we did intracellular staining to look at interferon alpha and TNF alpha, having gated on the plasmacytoid DCs. And you can see if there's no ex vivo stimulation, there's nothing being produced. However, if you stimulate these cells ex vivo with a cocktail of TLR ligands, what we call the viral cocktail, which is TLR7, TLR3 ligands, then you see very robust production of interferon alpha in the healthy PDCs in the healthy subjects, but markedly reduced production of interferon alpha in these COVID-infected subjects. Now, that's just one example, but I can show you many examples in which this is true. I mean, this is a highly significant result. Now, that is in the plasmacytoid dendritic cells. What about myeloid dendritic cells? Well, if you do the same experiment, take PBMCs from healthy or infected people without any stimulation, but now you're gating on the myeloid dendritic cells, there's hardly any cytokines you can see here. However, if you now stimulate with bacterial TLR ligands, which is TLR4, which senses lipopolysaccharide, TLR5, senses flagellin, TLR2, which senses peptidoglycan, you see there's a significant production of interleukin-6 and TNF-alpha by these myeloid dendritic cells, greatly diminished production in infected cells. Similarly, if you stimulate with the viral TLR ligands also, you see significant IL-6, TNF-alpha diminished in COVID-infected patients. And again, this is a highly significant result. You can see many donors here. So that was interesting. It showed us that in COVID-infected patients, dendritic cells, myeloid dendritic cells or plasmacytoid dendritic cells were impaired in their capacity to produce these cytokines. This was strange because there were reports emerging that there was increased level of cytokines, pro-inflammatory cytokines in the plasma or serum in COVID-infected patients. And indeed, that is what we found as well. When we did plasma cytokine analysis using Olink, we see increased interleukin-6, TNF-alpha, MCP3, and in fact, many of these cytokines correlate with the disease severity. So in blue are patients in ICU, red, severe patients, moderate, healthy patients. So that was consistent with the published reports, but seemingly paradoxical with the fact that we're seeing the cells in the blood are not able to produce these cytokines very efficiently. I should also mention that we also discovered that there were 3 molecules, oncostatin M, TNF superfamily 14, also known as LIGHT, and NRAGE, which were highly expressed, particularly in patients who were critically ill or had severe disease. These molecules had not previously been shown in the context of COVID, and we were very interested to see that these were highly associated with disease severity, and it raised the prospect that these could be potential therapeutic targets for COVID. Particularly because LIGHT and NRAGE have previously been described to be associated with other kinds of lung inflammation, COPD and asthma and so on. So we were then delighted to see that just a few weeks ago, maybe 3 weeks ago, this company in America called Seroco have announced the interim results of a clinical trial that they had done. They had an antibody that can block this molecule, LIGHT. So they have an anti-LIGHT monoclonal antibody, which they used in severely ill COVID patients to try and see if there was any therapeutic benefit. And indeed, the indications of this initial clinical trial does suggest that in severely ill patients, particularly patients above the age of 60, there was a very significant effect of blockade of LIGHT. So that's very interesting, and hopefully this will now be confirmed in larger clinical trials. Okay, now I come back to the fact that in the cells, in the dendritic cells, we were unable to induce efficient production of cytokines in vitro. To understand why this was the case, what we did was this technique called CytSeq. Many of you may know CytSeq is a single cell transcriptomics method in which we can analyze the gene expression profile in individual cells, but it combines antibody staining of cell types with single cell transcriptomics. And using this method, we analyzed the single cell transcriptomics of 9 COVID patients, 3 severe, 1 ICU, 3 moderate, 2 convalescent, 5 healthy, and 2 patients with influenza A or 2 patients with RSV. And you can see in this UMAP, and using the CytSeq method, you can very nicely define all the clusters of immune cells in a drop of blood. You can see T cells, NK cells, PDCs, red blood cells, and so on. And if you look at the transcriptional profile at a very high level, you can see that in fact, many of these clusters have gene expression patterns that are altered in response to COVID-19 infection. And in fact, flu, for example, has certain distinct clusters that seem different from COVID-19. Now, if you then say, well, what about severe disease versus moderate disease? How are the transcriptional profiles different? You can see that in patients with severe disease, shown here in orange, there seems to be distinct clusters in which there's a transcriptional activity relative to what you see in moderate patients in purple or convalescent patients in green. Now, the first question was, Can we find evidence for interferon alpha or interferon beta in these cells? And you remember I showed you that if you stimulate these cells with TLR ligands, you didn't see much interferon alpha. Even without stimulation, there was absolutely no evidence of interferon alpha. And to confirm that, what we saw as in the CiteSeq transcriptional data, there was really no evidence of interferon alpha beta. However, we were surprised to see that interferon stimulated genes such as IFI27, IFITM3 were expressed quite a bit in the CiteSeq data. Now that was a puzzle. How can you have interferon stimulated genes when you do not have interferon alpha or beta? Well, we looked at this more carefully. We looked at each cluster. So this is one cluster, CDC2, classical dendritic cell 2 cluster. And we looked at all these interferon stimulated genes here, IFI27, EIF2AK2. And you can see that these patients in purple, remember, these are the moderately infected patients with moderate symptoms. There's significant expression of many of these ISGs. However, in patients with severe disease in red, there's much less expression of ISGs. And of course, in healthy subjects in black, there's hardly any. And in convalescent subjects, there's hardly any. That's one cluster. Similar profile in the PDC cluster or plasmacytoid DC cluster. So it would appear that these ISGs are expressed chiefly in subjects who have a moderate disease. Now, we wanted to confirm this because this is an important point. So we did this analysis now with a larger number of subjects, 21 people, and we did bulk RNA-seq analysis. And here, what we saw was that, yes, indeed, there's expression of all these interferon-stimulated genes. And indeed, this seemed to be confined to moderate subjects, although it wasn't perfect. Some of these severe subjects also had this. So what we did was a variance analysis to see what variants could explain this pattern of interferon-stimulated gene expression in this data set. Was it age? Age didn't contribute very much. Was it gender? Gender didn't contribute very much. Was it disease severity? Well, it did have a significant contribution. But look, the time post-symptom onset had the largest contributing effect to this ISG profile. So we thought, well, maybe when these patients acquire symptoms early after symptoms within the first week or so, maybe that is when we would see this interferon stimulated genes. But why would they be expressed? Because if there's no interferon alpha, or is there interferon alpha? So we decided to measure interferon alpha, and we did this using this technique called SIMOA, which is an ultra-sensitive technique that can measure femtogram per milliliter quantities. Remember, 1,000 femtogram is 1 picogram. So that is 1 picogram. And you can see this is days post-disease onset, and you can see that there's this initial burst of interferon alpha that is produced. It seems to peak at around 1 week or 10 days or so, progressively declines. So that we thought, well, that's interesting because we saw the interferon stimulated genes also being expressed quite early after disease onset. So what does it look like if you correlate the amount of interferon alpha you measured by SIMOA in the plasma versus the interferon stimulated gene score that we measured by CiteSeq? And you see this beautiful correlation. So this would suggest that there's an early but transient burst of interferon alpha in response to COVID, and this then switches on these interferon stimulated genes. Now, subsequent to our paper, or I should say 2 weeks before, about 10 days before our paper was published, this beautiful paper by the group of Benjamin Tarrier in Paris also showed that in fact in COVID patients there was diminished production of interferon alpha, particularly in critically ill patients and severe patients, and consistent with that, diminished interferon stimulated gene score. Subsequent to the publication of this paper and our paper, this beautiful paper from the group of Adrian Hayday also showed that in fact in COVID patients, there was a time-dependent decrease in the amount of interferon alpha detected in the plasma. You see, this is days post-disease onset. In some of these patients, they have 2 time points, high early, and you see it goes down with time, high early, goes down with time, high early, goes down with time. So it seems, and then there were many papers. In fact, this is another paper showing there was inhibition of interferon alpha production in the PBMCs of COVID-infected patients. And this was one of the earlier papers. But in fact, there are 1 or 2 papers that also show that with increasing disease severity, there's higher levels of interferon alpha. So this highlights the need for there to be longitudinal analysis of having multiple time points from the same patients to actually arrive at greater consensus on the pattern of interferon alpha and ISG expression. The other thing that we discovered in our CITESEQ data was that, in fact, the expression of HLA-DR was diminished in these classical dendritic cells and classical monocytes, particularly in severe patients. That was consistent with the CITOF data. In fact, there's lower levels of HLA-DR in the myeloid DCs and classical monocytes. And in fact, this was highly correlated with the expression of this molecule known as S100A12 that was confined to this cluster of monocytes or myeloid DCs. What is S100A12? Guess what? It is the same molecule that we had measured by Olink called ENRAGE. You remember that from my few slides ago. And you see the expression profile of S100A12 or ENRAGE is inversely correlated with the expression of HLA-DR. So the more ENRAGE these monocytes or myeloid DCs express, the lower HLA-DR they express, and that makes sense. Severe patients express very low levels of HLA-DR, and they express very high levels of ENRAGE. And in fact, the ENRAGE data that we measured by CITESEQ and by Olink correlate very nicely. Finally, what we also saw, and surprisingly, and we were wondering how could you explain this? A lung infection, virus in the lung is resulting all these dramatic effects in the blood. Could there be virus in the blood? Well, we could find no evidence for virus in the blood. What else could it be? Well, we thought maybe the lung is not a sterile environment. It has bacterial products. So can we look at bacterial products such as lipopolysaccharide or bacterial DNA? And in fact, in the plasma of these patients who had severe disease or critically ill, there were significantly higher levels of bacterial endotoxin and bacterial DNA in the plasma. And in fact, the amount of bacterial DNA on the x-axis correlates very beautifully with the cytokine, pro-inflammatory cytokines, whether interleukin-6, TNF, MCP3, LIGHT, oncostatin M, ENRAGE, arguing that these bacterial products may have an important role in mediating the cytokine production, excessive cytokine production that is seen with COVID patients. So let me summarize at this point. I think these data point to the following model of what might be happening in COVID-infected patients. Early, when I say early, it's early disease post-symptom onset, maybe 7 days or 10 days. There's an initial burst of interferon alpha. We believe this is coming from the lung. And this initial burst results in switching on of these interferon-stimulated genes, which were detected very early after infection, after, I should say, disease onset. However, in patients with late post-symptom onset, there was excessive inflammation, evidenced by the production of inflammatory cytokines such as TNF, IL-6, LIGHT, ENRAGE, and so on in the plasma, bacterial products. And we think this is coming from the lung because we could not get the cells in the blood to make these cytokines, except ENRAGE that was detected in monocytes. And in the periphery, in the blood, as you saw, there was diminished production of type 1 interferon in plasmacytoid DCs, diminished production of innate cytokines in monocytes and myeloid DCs, and the emergence of this population of HLA-DR low ENRAGE expressing cell, which previously, in the context of bacterial sepsis, has been described to be a kind of myeloid-derived suppressor cell. Now, the question is, how can this be so? Why are you having so much excessive inflammation in the lung with all these cytokines, but far away in the blood, there seems to be impairment of the innate response? What is the connection between the two? So to understand this connection, what we did was a multi-scale, multi-omics analysis, not only using CyTOF and RNA-seq and Olink, but now we also added metabolomics, because we hypothesize, well, if there's no virus in the blood and there's bacterial products, what else could be? What else could it be that mediates this link between the tissue and the blood? And we suspected it might be metabolites. So to do this, we adopted this multi-omics approach, which we had used in the context of vaccination in the past, where we can integrate data sets from transcriptomics, metabolomics, cytokines, CyTOF or mass cytometry, and computationally integrate this sort of data analysis. And I'm going to show you just 3 or 4 exciting pieces of data from that. When we did the metabolomics, one thing that we saw that was amazing and quite striking was that the concentration of the amino acid tryptophan was reduced in COVID-infected patients. Here we are, you can measure tryptophan here. This is in healthy people, moderate people, a little bit less, severe people, a little bit less. But look, ICU patients, very substantially reduced. Now, what is tryptophan? It's an amino acid. How is it metabolized? It's metabolized by these enzymes, IDO1, IDO2. And how are these enzymes regulated? They are regulated by inflammation. Bacteria, viruses, inflammatory cytokines, all of these can activate these enzymes. These enzymes result in the breakdown of tryptophan into N-formyl kynurenine, and this results in kynurenine. Now, okay, so if its tryptophan is catabolized, what happens to kynurenine? It should go up. And is that so? Well, that is so. If you look at kynurenine here, moderate, severe, ICU is up. Okay, kynurenine then gives rise to kynurenate. That should go up. That also goes up. That should give rise to quinolinic acid, and that should go up. Yup, that also goes up. So this whole pathway of tryptophan metabolism is activated in COVID patients. But what about these enzymes? Are they expressed in COVID-infected patients in their cells? And we did this using flow cytometry. I'm showing you the data from myeloid dendritic cell. Look, significantly increased expression of IDO in the myeloid dendritic cells of COVID-infected patients. Now, I told you tryptophan is reduced. Now, tryptophan is an amino acid. How does the immune system, or how does the body sense amino acids? One of the major sensors of amino acids is this molecule called GCN2. GCN2 is one of 4 different molecules that can sense different kinds of cell stresses. GCN2 senses amino acid deprivation. This results in the phosphorylation of eIF2 alpha that shuts down translation of messenger RNA in ribosomes. But certain types of messenger RNA can be translated, and these are messenger RNAs involved in the stress response, and they are transcribed and translated, and transcription is dependent on a transcription factor called ATF4. ATF4 is one of the major transcription factors that mediates downstream the effective functions of GCN2. So we said, all right, if there is tryptophan deprivation, GCN2 must be activated, and we found that was the case. And if that is activated, then ATF4 must be activated, and the downstream targets of ATF4 must be induced. And we looked at the downstream targets of ATF4.
We have 1 minute left.
So. Sure. This is the last slide. We looked at this, and in fact, there was a dramatic change in the expression of these ATF4 target genes. So this is the model that we believe is happening, that in these COVID-infected patients, there's a massive perturbation of the metabolites. There is, in fact, deprivation of IDO that results in, actually this induction of IDO that results in tryptophan catabolism that then activates GCN2 and these ATF4 target genes, which then mediate this immunosuppressive phenotypes. And so the question is, what does this mean in terms of infections? Are these patients susceptible to other kinds of infections because there's impairment of innate immunity? And indeed, we got PBMCs from these cells, infected them with dengue or Zika, and you can see there's enhanced replication of dengue and Zika. And this seems to correlate with the amount of interferon alpha that's produced. So in other words, the less the capacity to produce interferon alpha, the more susceptible these cells are to dengue and Zika infection. So that's all I have to say. I kind of rushed through the last bit, apologize. These are all the amazing people who did the work. Prabhu Arunachalam, Florian Wimmers, and my lab were the main people who led the work in collaboration with these other labs who I mentioned. Thank you very much.
Thank you so much, Bali. We have maybe time for one question, so I would like to briefly ask you about ADE. We hear a lot in the last days about antibody-dependent enhancement, and you mentioned dengue. And so I was wondering if any of your findings could be related to that phenomenon, if it does exist.
Yeah, I mean, I think there is ADE that's been suspected. We haven't looked at this specifically in our cohorts, but you know, there is certainly with SARS-1, the initial SARS, this has been reported, particularly with vaccines. We haven't seen any evidence of ADE, and we haven't been looking for this, but it's something that people should look for, either in infection or in the context of vaccines as well.
Okay, thank you very much. And if we have any further questions, you can pick them up from the Q&A chat. And I'm delighted to move on to our next speaker, last but not least, Sharon Peacock. Do we have Sharon with us? Great. So,
yeah, yes, I'm here. Yes, I'm just getting myself sorted out. I'm hoping that you can see my slides.
Yes, we can see your slides.
Can you see them? Are they in full full screen mode, or do I need to do that?
Yes, excellent.
Does that work for you?
Yes,
Fabulous.
good. So just to briefly introduce Sharon Peacock, who's a professor of public health and microbiology in the UK, in Cambridge. She's been working for many years on microbial genomics, and is now very much involved in the genomics of SARS-CoV-2, also as the director and lead for COG UK. So Sharon, thank you very much for joining us, and the topic would obviously be tracking the variants through genomics.
Thank you very much. Can you hear me okay? Is everything okay? Good, great, thank you. Well, it's an absolute pleasure to be here. It's quite busy at the moment, but it's so wonderful to stop for a moment and talk to you about some of the work that we're doing in COG UK. So as you heard, I direct COG UK, and what we're doing is using large-scale viral sequencing to better understand the virus and then control it. So I'm going to try and put my slide on. Yep, there we go. We're getting the hang of this. So let me tell you a bit about COG UK in the first instance. So back in March, when there was about 80 cases cumulatively in the United Kingdom, a group of academics got together and decided that they were going to put together a consortium to sequence SARS-CoV-2. Now, at the time, we anticipated that we would need to sequence about 180,000 viral genomes. I thought that was quite a lot at the time. We've actually subsequently sequenced 279,000 so far. But an incredible group of people got together middle of March, and we were given a room at the Wellcome Trust in London, and we worked out a blueprint for how we were going to set up a network for COVID-19 sequencing. Now, we started on the 1st of April, and this, this slide shows where our sequencing sites are. So we've got 16 regional sequencing sites, and unusually for something that's effectively providing service to the country, most of these sites are actually academic sites. So basically we scooped up into one place everybody who could do sequencing and put them into COG UK. So across the United Kingdom, we have numerous academic universities. We also have the 4 public health agencies of the United Kingdom. We also have the Wellcome Sanger Institute, our big sequencing capability in the country. Now, we were very fortunate because by the 1st of April, our government had funded us to the tune of £20 million, and I would like to thank Sir Patrick Vallance for his support in that. And we also have funding from the Wellcome, and the project is administered in Cambridge with myself as director, but as a very large team of people to help me. Now, what do we do? Well, we really have 4 aims in our consortium. The first is that we want to detect mutations and variants. This is so important to us at the moment, and I'll be talking quite a lot more about mutations and variants later on. In fact, most of my talk is going to be on that. We also use sequence data to track the virus and its spread to support outbreaks. So we've used it in a very large number of settings, including universities, schools, hospitals, care homes, factories, etc., to try and understand how the virus is spread. The third thing we do with our data set is we combine it with other data sets, including human sequencing. And so we have a large human sequencing program in the United Kingdom, and we're actually busy combining that data at the moment. So we have a host of viral data set. We're also combining that with our phenotypic work and our immunology work. So one of the things that the pandemic has really done in the UK is galvanized a collaboration across the country. Finally, we also support national and international collaboration. So we're in the process of looking at how we can develop an online learning environment to allow other people to access what we do. We also give everything away. It's a very giveaway culture. So we make all of our protocols and our methods available to other people. We upload all of our data that we generate into GISAID. So when we think about what our sampling strategy is, we have 3 prongs, if you like. First is half of all the sequencing we do is completely unbiased, and it's from hospital patients and from the community. The reason we do that is because we think that that gives us the maximum chance of picking up new variants as they arise. The second is that as we detect new variants, we do targeted sampling. So we have surge testing around variants of concern, and we do sequencing as a result of any positives. We also have national core studies, and that's a wide range of studies that relate to reinfection, vaccine failure, and immunology. So on the very right-hand side, you'll see our coverage. We produce a coverage map every week, which goes on our website. So if you're interested, you can see how we're doing. And we're aiming to get between 5 and 10% coverage at the moment. That's quite a tough ask because the UK has got a lot of cases of COVID-19, unfortunately, at the moment. And as the prevalence of the disease comes down, our coverage will go up. And so I'd really like to see 50% or more of our cases being sequenced. So you've got the total number there, nearly 280,000 viruses sequenced to date. Let's see if I can progress the slide. There we go. So I'm going to really talk for the rest of my talk about SARS-CoV-2 mutations. And what I'd like to say is that I spend a lot of time talking with the public and with the media about what these mutations actually mean. And I'm trying to develop a narrative around the fact that much of this needs to be normalized. There's some that are really quite concerning and to be focused on, but mutations are occurring all the time. So I'm sure that you're very aware that SARS-CoV-2 is mutating at around 1 or 2 times a month. Now, of course, people say that's a very low rate of mutation compared with something like influenza. I think the difference to that is that when you have 100 million infections on the planet, and probably a lot more than that, every time the virus goes through an infection cycle, it has the opportunity to make a mistake in its genome. And so although it has a very low rate of mutation compared to other viruses, it certainly has an awful lot of opportunity to mutate. And we're getting to a point now where we're getting a combination of very high infection rates together with increasing immunity in our population, either because of natural infection or vaccination, which is a heady mix for natural selection. But when we think about mutations, actually there's three outcomes for the virus. First of all, the overwhelming majority of mutations have no effect on the biology of the virus, and it's no apparent change. That will certainly allow the virus for us to track the virus, because you could develop a kind of like a genetic barcode, if you like, so you can see the differences between the viruses, and that develops into lineages. That's very useful for tracking the virus. Now, some mutations are going to be disadvantageous, in which case we're unlikely to see them, or they'll become extinct. But the thing I really want to focus on is those mutations which have an advantage to the virus, that provides a fitness advantage to the virus, that may result in lineage expansion. Now, this is the four areas that I think about. I use the word tilt. It just reminds me of what I should be thinking about every day when I think about mutations. So the mutations I worry about in particular are those that, of course, are going to lead to a change in the amino acid, and that could lead to a change in the structure and the function of the virus. So these are the ones we're focused on. And they could lead to four possible changes of phenotype. And there could be one phenotype, or there could be a combination in the same virus. So does a mutation increase transmissibility? Does it change the way the virus interacts with our immunity, so that we have reduced immunity against the virus? Does it change the virulence or lethality of that virus? And finally, does it alter our ability to test for SARS-CoV-2? And what we've seen actually is all four are beginning to crop up right now. Now, I'd like to just start with a couple of examples which really sets the scene for kind of mutation hunting and trying to make sense of what mutations actually mean. So I've got a couple that have been under past surveillance by us. The first is a mutation in the spike called D614G. That's a replacement which wasn't present in the virus when the virus first emerged, but it's now ubiquitous across the world. So if you look at the graph right on the right there, the top right-hand corner, this is data for UK genomes. And what you'll see is it's a number of samples over epidemic week along the x-axis. So if you look at wild type, which is the virus that first emerged, you'll see that in the orange, and the D614G is in the green. So you'll see that certainly in the UK, and actually very much in the world, D614G became spread across the world. Now, why did it do that? It certainly suggests that it has a selective advantage, and the replacement was associated with higher viral loads and younger patient age. But it sort of went largely under the radar in many ways, because although it was interesting at the time, there was no association with greater disease severity. So if you look at the graph there at the bottom right-hand corner, what you'll see is outcomes of people who went into hospital and were infected with either the wild type or the G version of the virus. So you'll see that it's broken up by age groups and divided into the D or the G version, and no respiratory support, whether someone needed oxygen or whether they needed a ventilation or they died. And based on that, we concluded that the 614G was not more lethal or virulent than wild type. So the next particular variant that really is under past surveillance was something that emerged in the summer over in Spain. This is a very elegant work done by Emma Hodcroft. And in the map at the bottom, you'll see that after emerging in Spain, this particular variant spread across Europe very rapidly. Now, at the time, Emma proposed that it could be that there was a mutation, a substitution called A222V in the spike protein. And actually what we did was we looked at the prevalence of this particular variant over time in the United Kingdom. So if you look again at the top right-hand corner, we have a particular piece of software which is tracking our mutations, which actually is going to go live probably towards the end of the week, so you can have a look at it. And you'll see here that the wild type, the A222 is in green, and the variant is in orange here. And actually, if you were to stop your investigation by about week 46 or 47, you'd think that there was something rather important about this particular variant. But actually what we found was over time it kind of fizzled out. There was no increase in virulence. And actually what we concluded that this was spread through summer travel. Now that sets a problem for us, because if we're looking at the importance of a variant and you get this much signal with something that actually doesn't prove to be important in the end, you then have an issue over deciding, looking in the needle in a haystack to say what is important and what isn't. So I'm coming now to something that you'll be very familiar with, B117, first detected in the UK, called Kent variant or new variant, but I try to avoid using particular country names. And so 117 was first detected in the south of England, and looking back, it was in the data set from as early as the 20th September last year. It was just on its own. It didn't look particularly interesting. Nobody noticed it. But since then, it has spread across the UK. So it accounts for about 90% of all of our disease. And it's also been reported in 80 different countries. So you'll see in the top right there, the map of its spread across the world. And this is one to watch out for. The reason I say that is because the estimates of increased transmissibility goes between about 35 and 70%, but it has spread like wildfire in the United Kingdom. There is a question mark over whether it's more virulent, but increasingly our data indicates that it is. Now, the data in particular shows that once someone's in hospital, there is no increased risk of death, but actually there's a higher risk of going into hospital, i.e. severity of disease is greater with this variant than with other variants. But fortunately, once you get to hospital, then you have the same survival outcome as other people. Now, this also affects our diagnostic tests, because there's a deletion in the spike that actually leads to S-gene target failure in some tests. Now, fortunately, that doesn't lead to a negative test because we have 2 other targets in those tests, but it's also used as a useful surrogate because our mega labs use a particular assay where we have S-gene target failure. So we can track it as a surrogate. Overall, we think that immunity is largely unaffected. There's some minor noise in neutralization assays. The problem is that it's now acquiring a mutation, which we refer to as E484K, and this, unfortunately, is associated with reduced immunity. So this is one to watch out for. It has a combination of mutations that are particularly troublesome. And in fact, this has been a fascinating variant to think about, because if you look at the phylogenetic tree on the right-hand side, and you'll see in green the isolates of 117 on the phylogenetic tree, what you'll notice is that this particular cluster stands out markedly from everything else. And in fact, that's one of the first things we noticed. It has striking genetics in that it has 23 different mutations compared with the wild type. And 14 of those are amino acid replacements with 3 deletions. Now, the challenge for us, you've seen the previous slides, the challenge for us is when you call something important. So we spotted this in the middle of November when it started to expand quite rapidly in our population. In Kent, there was a big surge of cases, despite lockdown, and everywhere else cases were reducing, but in Kent this was surging. And it was when we put the epidemiology together with the genetics that we concluded that this was significant. But actually calling that out is actually more challenging than it sounds. Now, just to say that one of the amino acid replacements, N501Y, sits in the receptor binding motif of spike, and that increases binding affinity to the human ACE2 receptor. So this, as I say, is one to watch out for, and I know that many other countries are concerned for its egress out to other parts of the world. Now, my next variant of interest, and this I think is the one that I'm particularly concerned about, is what I call B351. And please forgive me if you use a different nomenclature. There are several that are used, but this was first detected in South Africa. Now, this is now in 40 countries, you can see in the map, and it appears to be more transmissible. Immunity is affected, and so it's likely that vaccination is likely to be effective, but only partially effective. And we don't yet know in full about its virulence potential. Again, it's got some spike gene mutations of likely significance. You'll see there it has the N501Y, which is we associate with transmissibility. It has the E484K, which we associate with reduced evasion of the immune system. So my next slide really looks at the frequency of the South Africa variant, or the first detected in South Africa. And what I'd like to do here is this graph is interesting because it shows the number of sequences produced since the first new variant was reported per country. So some countries are sequencing very little because of insufficient capabilities. But if you look at this, what you see is that in Zambia, for example, even though there's only 31 genomes in the GISAID database, 33, beg your pardon, 31 of those are this particular variant. And if you go along the graph, you'll see that 37 and 45 from Botswana, about half from South Africa, and about half from Mozambique. And so the number of genomes in GISAID is not a good, accurate representation of what's circulating. But what that tells me is that certainly around South Africa, there's been a substantial, likely to have been substantial transmission. Now, my last variant that I'm just going to refer to is called P1, first detected in Brazil. Now, this seems to be less spread to just 18 countries. Again, it's got a characteristics in its genetics that make it more likely to be more transmissible, to affect immunity, and we're not clear about virulence. You can see that the protein substitutions there, the same as the variant detected in South Africa, but actually quite low numbers at the moment. So it doesn't seem to be roaring away right now. So what I'd like to do in my last slide, yep, here we go, is really start to talk about how we think about these variants, because in the last probably 2 weeks, we've seen quite a lot of different variants arising coming into the United Kingdom, which actually also have some concerning mutations, particularly E484K. So we have something that's come in that's an international lineage called A231, which has actually developed the E484K. Our own homegrown lineage now has developed E484K. And every day there seems to be somebody ringing me up to say there's a new lineage with E484K. And so rather than thinking about lineages all the time, the way that we're thinking now is to start to think about a convergence of mutations. So convergent evolution, because all around the world, the same mutations are cropping up again and again. You can see here a distillation of that, certainly for the 3 lineages that I've mentioned. And you'll read across to see that the same mutations are coming up, which clearly are associated with a natural selection and emergence in the face of very high disease burden and a rising immunity in our population. So this particular table, we're expanding now to really say, how do we get to grips with this and actually tweak our vaccines? We're going to need to have to tweak vaccinations, almost certainly, or vaccine developers are already beginning to change their vaccines. It's very likely over time that we're going to need to have booster doses for our population to vaccinate against the variants that are circulating at the time. So really just to finish up, to say that I think that we're at the very beginning of a journey of the emergence of variants and mutations. It's possible that there'd be a relatively small number because of convergent evolution and the emergence of particular mutations that are associated with a fitness advantage relating to those particularly the 3 things I've mentioned. In fact, the 2 really, transmissibility and immunity. There's a lot of debate about whether lethality does or does not confer a fitness advantage on the virus. I suspect that that's not the case, but we could debate that. But what we're trying to do now is get our head around the leaderboard of what are the top 10 or the top 20 mutations that occur that lead to an amino acid replacement that we really need to try and target our vaccines against, because this is going to be an ongoing cat and mouse game with the virus. Actually, I do have one more slide. I've already said most of this, that I think there's going to be an ongoing emergence of undesirable variants driven by transmission and by partial immunity. And that makes it very complex to track. So, for example, we're thinking about constellations because if you think about the UK variant that's now developing more mutations on top of the existing mutations. So what are we going to need to do? Well, I certainly think that we're going to need a global network of sequencing capabilities and data platforms. I know that many countries are looking to scale up their own sequencing capability. And it's only when we've got eyes on the virus that we'll actually be able to understand what's circulating in our own populations and how to best protect them. Data sharing is going to be really important with global information being shared across the world. We're going to need to look very carefully, of course, at a pipeline for phenotypic tests in immunology, so that if a mutation is very common, what does it actually mean in terms of its biological importance? And we're going to need to have to monitor the distribution of the variants globally, and then continue to tweak our vaccinations. So just to finish then, this consortium is around 400 people. They work across the country in the United Kingdom. We're having increasing international collaborations now and are developing networks across the world. But I just want to say a massive thank you to so many people that have made what we do possible. So thank you very much, and I hope you have some questions.
Thank you so much for this splendid talk. I don't see we have any questions in the chat yet, so maybe I'll start off with a question. There has been a lot of discussion regarding travel restrictions and the possibility of containing variants or preventing importation of variants via travel. And I wonder, what is your view about this in a global scale?
Well, I can tell you what we're doing in the UK, and then perhaps I could reflect more widely on that. So in the UK, we do have quarantine now from 33 what we call red countries. So people need to go into hotels for a period of time, and people travelling from outside of those countries, they need to go into home quarantine. And we're sampling people twice a week when they're in hotel quarantine to detect what they may be infected with. Now, I think that's the right thing to do. I think that the variant first detected in South Africa is a real risk and a real threat to vaccination. But the second prong is that if we spot this in our population, then we do try to do enhanced contact tracing, swabbing with surge testing. But there's another part to the story, and that is that our common or garden variant now, the B117, is beginning to get the E484K variant. And so there's a point in time when it's possible to lose the battle, and in which case what we all need to be doing is really controlling our borders as appropriate for our country, but actually driving down the number of cases, because if you haven't got infection, you don't have mutation. So the best possible way of reducing mutation is actually to reduce case burden.
Thank you. We have a question. Do you see any evidence that the variant that emerged in South Africa has any chance of taking hold in the UK, or does the UK variant appear to be more transmissible?
That's a really good question. Well, in South Africa, that variant appeared to transmit quite rapidly and widely, and that was an important observation in South Africa, and it was infecting people who had already apparently been infected. Our current experience is that we know of only around 200 cases. There's probably more, but it doesn't appear to be highly transmissible at the moment. I think that has to be judged a few weeks down the line, but it doesn't appear to be spreading like wildfire, as it were. It's relatively contained at the moment, but it's early days. So whether it will outcompete our circulating virus or not is another matter. It doesn't appear to be at the moment. And with our kind of background virus of 70% or so more transmissible, 50% more transmissible, you'd have to be quite fit to overcome that. So let's see what happens.
Thank you. We have time for maybe one last question, and that question pertains to diagnostic pipelines and how the continuous emergence of variants could or should impact the development of diagnostics.
Well, what we're doing is we have a constant monitoring system for when diagnostic tests don't appear to be working as we might think. So we noticed the S gene target failure. We've noticed that other targets are failing sometimes, and what we do then is we sequence those particular viruses to find out why. And it's not altogether uncommon to see deletions or changes in the genetic code leading to a diagnostic failure. So we're on the lookout the whole time, and we have a group that looks at diagnostic performance. At the moment, I don't think it's a massive problem, I have to say. I think that the diagnostics, we can be confident that they work. But the other question for me around diagnostics is actually, can we be using PCR-based assays to detect mutations of particular concern? And we're evaluating those at the moment. They are in use in other countries. So for example, can you use an assay to detect E484K? I think that's all well and good, but what that doesn't do, of course, is pick up new variants and things that you don't know about. So I think that both probably have a role to play as we go forward in controlling this pandemic.
Thank you very much. And on behalf of Maurizio, my co-chair, and myself, I would like to thank all four speakers for really a great symposium, and also to thank the ESCMID office for organizing everything. And I look forward to seeing you all in our next webinars, and of course, ECCMID in July. Thank you so much, and have a good evening.
Thank you very much. Bye-bye.
Bye-bye.